Hi guys, welcome today for the today's section of membrane transport. Right, molecules they are to be taken inside, they are to be thrown outside, the nutrients are to be absorbed, the waste products are to be given down. This is what something is to be done by every cell. Right, today we are just going to have an overview of by different ways by which a cell it does this. Right, आपको कोई चीज अंदर लेनी है, कोई चीज बाहर डालनी है सेल के, इसमें आपको मेजर कोई बैरियर होगा तो that is your cell membrane. And cell does it in a very elegant way, in a very efficient way, and by different mechanisms it achieves this particular purpose of transporting the molecules in and out. So basically all transport systems we classify into two basic types, right? One is what is called as an active transport and other is called as a passive transport. Be with me. Active transport requires energy. Remember, passive transport does not require energy. Passive transport will transport the molecules from higher to lower concentration. So if you are going from high to low, obviously the flow is natural, it is in law with thermodynamics and therefore it does not require energy. So such type of transport which is down the concentration gradient, remember, which does not require input of energy, which is going from higher to lower, is a passive transport. A transport where you are going from lower to higher, that is against the concentration gradient, you are going to require energy, and that is the active transport. Let's have an overview, a simplified version. Like this, you can see a typical membrane I have shown. You have a first mechanism, that is simple diffusion. Movement of molecules from higher to lower concentration is what is called as your diffusion. So when molecules are spreading out, diffusion literally means to spread out. So whatever molecules are allowed by this particular membrane, if they are at a higher concentration here and a lower concentration here, they will move in, right, if they are allowed, obviously. So that particular phenomenon where no mediator is involved, no energy is involved, molecules are just moving, this is possible for very small molecules which can cross this lipid bilayer, which are non-polar. This becomes very difficult for a charged molecule, for a large molecule, but for these simple, some of the gases and some simple molecules, this process works. So that is simple diffusion. So simple diffusion, movement from high to low, no protein involved. Then we have another what we call as facilitated diffusion. As you can see, there is no energy involved here, so these are passive. So diffusion comes under passive transport. This facilitated diffusion also comes under passive transport, but only thing is that the molecule cannot travel, maybe because it is charged, the non-polar membrane does not allow. So if you have a carrier molecule here which is allowing the passage of the molecules, that is what is called as a facilitated diffusion. So facilitated diffusion, similar to this, going from higher to lower, down the concentration gradient, working till there is equilibrium on both the sides. एक बार दोनों side concentration same हो गई, it will stop. So this particular facilitated diffusion is brought about these carrier protein molecules without expenditure of energy from higher to lower concentration. So that is facilitated. It shows three basic characters you might be knowing. It is specific. So this protein will be specific for the molecules to be transported. It can be inhibited by specific inhibitors, and this transporter it can be saturated. So these are what we call as the characters of a typical facilitated diffusion. Now coming to an active transport, where most of the times you have to carry the molecules from lower to higher concentration. So for that movement you require expenditure of energy in the form of ATP or like any other molecule which can provide you energy. So when you hydrolyze ATP, release the energy, use the released energy for pulling in the molecule from lower to higher concentration, that is what is referred to as a primary active transport. So this what you can see is a typical primary active transport. So in a typical primary active transport, molecules are taken from low to high against the concentration gradient by using the hydrolysis of ATP as a source of energy. Sometimes a particular ion is moving down the concentration gradient. You have first created a gradient, so there was an expenditure of energy to put an ion outside. So now concentration of this ion outside is high, inside is less. So ion moves from outside to inside. The molecule which you want to take in, that is at a lower concentration outside and at a higher inside, but still you want to take in. So when this ion will move in, it releases energy which can be used for this particular solute to be taken from outside to inside, that is from its lower concentration to higher concentration. So such type of transport we call as a secondary active transport. Remember, this also requires energy. This is also against the concentration gradient, but it uses the gradient of this ion. obviously which you created earlier. We'll see this in detail in some other mechanisms. So secondary active transport, movement of one molecule accompanied by the other molecule and taking it inside or outside, right, depending on the situation, is a secondary active transport. Sometimes there are channels, right, which move these molecules from what you call as outside to inside or inside to outside, which are gated. They may be gated by ligands, they may be gated by ions. So once you open this channel, molecules like they just rush in or they rush out, depending on where the concentration is more or less. So that is what is called as your ion channels. So you have different what you call as your ion channels through which the molecule moves. Then we have some molecules which we call as inophores, right? Molecules which are involved in transport of small ions from their higher to lower concentration. They are referred to as your inophores. Examples you can say your gramicidin, nigrisin, valinomycin. Specifically they are specific for ions like K+, Na+, like other ions. So these particular molecules they are referred to as your inophores. One more concept comes in when we are talking about your primary active transport is what is called as your uniport. You must have heard of. You have synport and you have what is called as your antiport. So three basic types. These two come under co-transport. So if you have a membrane, say for example, and in this particular membrane, if one molecule is moving from what you call as in to out or out to in, then that is what is called as your uniport. So only one molecule has moved in, that is what is called as your uniport. If you have a say carrier molecule, here also you may have a carrier molecule, no doubt. Here if you have a carrier molecule and the two molecules they are moving now in the same direction together compulsorily. So if two molecules move in the same direction, we call this as your symport. So movement of two molecules in the same direction is called as your symport. Now if the molecules they are moving in opposite direction, again it is a compulsory phenomenon, one molecule has to go out, other has to come in, then such kind of movement is called as your antiport. Right, as I told, this is just a brief overview of different transport mechanisms. We are going to discuss each of them in detail in the upcoming videos. So this is an overview of different transport mechanisms which are working for taking the molecules in and giving the molecules out. Stay tuned with me, Professor Girish Kukreja, for more such insights into metabolism and microbiology in general. Thank you.